No. Okay.
Okay. Last but not least, Christa Martin PhD. She's the director of cytogenetics and an associate professor of pediatrics at Emory. Her topic is copy number variation in autism spectrum disorders.
Okay, and I promise I think I can finish by 12, so we're still out here on time and everybody can get home and enjoy the weekend. So first I'd just like to actually start by reminding everybody that really copy number variations are really just chromosome abnormalities in disguise, at least most of the ones that are talked about in the literature today. I think we're moving toward a trend of people looking at smaller and smaller and smaller variations in the genome. But what I'm going to discuss today pretty much are all categorized as just new microdeletion or microduplication syndromes. Copy number variation, I think, sounds more new and more exciting and helps us get manuscripts published and research grants funded, so it's become a very common term to be used in the literature. And I think we've seen this in several talks, but one thing I think is interesting I'd like to point out is, you hear the male to female ratio of 4:1, and that's sort of a general number in autism. But if you actually look at the difference between those children that have mental retardation in association with autism, the ratio really falls closer to 1:1 male to female, which you'd expect. And as you look at the higher functioning, the ratio actually becomes much more different, so more like 7:1 or 9:1, and these are studies that have been done by the CDC. The other important thing is most autism has other associated features. So MR is seen in about 50%. Seizures are another common feature seen in about 20 to 30%. And many people, as we've heard, have macrocephaly dysmorphic features, and GI issues are another common observance. And everybody has to show a pie chart, so I'm going to follow up with Brad's pie chart, but I think it's interesting to look at of everything that we know about autism. And again, autism is not one disorder, it's many. Most of what's known has been found out by single gene, or like Fragile X or UBE3A, P10, genetic disorders. There still exists this large component that people are saying is out there, that's this common autism, that could be a combination of multiple genes plus environmental factors, but that's really the big question mark still. The part we're starting to figure out are related to cytogenetic imbalances. And as Brad alluded to, there are several that are observed commonly, and by common it's about 1% or so of kids with autism. 15q maternally inherited duplications, 22q11 deletions of the DeGeorge region, 22q telomere deletions of the Phelan-McDermid syndrome, and 2q telomere deletions. And those were pretty well seen by a combination of either G-banding or FISH. But what we now know is through using higher resolution array analysis, we're finding that there's at least a 10% diagnostic yield in autisms by being able just to look at a higher resolution through copy number array analysis. And so today I'm going to try and focus on what's being done to solve the rest of that piece of the puzzle that's referred to as these autism spectrum disorders. And chromosome abnormalities are known to contribute about 5 to 12%, but if you add new array findings on that, we're already getting up to 20 or even more percent. And so I was just going to review some of the more common known cytogenetic causes of autism and go over some of the newer models and how we identify even yet undiscovered regions of the genome that contribute to autism. So this was a study published in 2005 by Borsman, where he reviewed the literature and any paper basically that mentioned autism and a chromosome abnormality. And you can see that every chromosome has been implicated in some way in autism. But as I just pointed out in the previous slide, there are several where you can see that the frequency is much higher than compared to other regions where they might just be individual case reports. One interesting thing is people are starting to look at these cytogenetic abnormalities and look at the phenotypes a little more closely. And in fact, in individuals who have DeGeorge syndrome, so this is just an example, FISH picture showing the DeGeorge deletion, people are starting to collect individuals with these deletions and ascertain them for ASDs. And what's coming out is that about 50% of individuals with the 22q1 deletion actually fall somewhere on the autism spectrum disorder scale, varying from autism up to even Asperger's syndrome. Another important category, and one I think is everybody needs to think about, are the 15q duplications. And this is the region that is, when deleted, is associated with Prader-Willi or Angelman syndrome. but when duplicated is associated with autism. And because this is an imprinted region, it's only associated with autism when maternally inherited. So a normal mother can carry this duplication. So here's the duplication interstitial on chromosome 15. But if she passes this to her children, male or female, they would be affected with autism. And so for a carrier female of this chromosomal constitution, her recurrence risk is 50%, which is quite high. And therefore, it would change the counseling and recurrence risk estimates for this family considerably. This can also be seen as a supernumerary marker chromosome. This just shows two pedigrees that were identified in the AGREE collection with this chromosome imbalance. Both of the mothers were shown to be carriers, and both families had 2 affected children. So here you see 1 female affected, 1 male, and this family had 2 males. We used to do this using FISH, now it's detected using microarray analysis. But another important thing I think to point out is that they really don't have identical phenotypes on the spectrum, so there's still some variability in their presentation. So in this particular family, the female was diagnosed with pervasive developmental delay, whereas the male was diagnosed with autism. Female had some verbal skills, and the male was really nonverbal and showed regression. So it's important that even though the phenotype doesn't match, we don't rule out some of these genetic causes because we know there's variation. And so how do we find new genetic causes or predispositions to autism? One of the ways that's been used and was alluded to in the previous talk is by trying to get large collections of samples of children with known diagnoses. And these are 2 efforts that have been put together to do that. AGREE was the first one of these efforts. It was created by the parent support group Cure Autism Now, and then actually was merged with Autism Speaks. And the goal of this resource was to collect multiplex families, so only families who had 2 or more affected children. But they go through a pretty extensive sort of Cadillac phenotyping to diagnose their autism and any other associated features that they have. So we were involved in this effort from the research side and looking at copy number changes and chromosomal imbalances. And I always found it interesting when we would go to walks or other family support groups and try to recruit families for this effort. The people that got really upset were the individuals who only had 1 child with autism because they were frustrated that there was no resource for them to be involved in research. And that's sort of where the next collection came out of. So there's a foundation in New York called the Simons Foundation, which does a lot with math and sciences, but now has gotten involved in the autism community. And when the Simons Foundation first approached a group of researchers and said, what is needed to speed autism research along? We all said, well, there's this great resource for multiplex families, but you're missing out on the majority of families with autism who only have 1 affected child. And so that created the Simons Simplex collection. And what they did was they set up 13 recruitment sites across the United States. And the reason to be included was families who have 1 affected child and 1 typical child, so that you could use that for comparative studies. This just shows the sites that are participating. So there's actually 1 in Nashville, and we have a site in Atlanta. A lot are in the Northeast, but then they're scattered on the West Coast as well. And the goal is to enroll 2,000 families that are simplex families, and they pretty much are brought into clinic and go through a 2-day battery of tests to really get a clear diagnosis of autism, look at their other language development, the parents are studied, the normal sibling is also studied. And so all of this information is being deposited in an online database that's then available to researchers. And they're also, in this study, they are collecting blood samples and making cell lines so that, like the AGREE study, these DNA samples are available to any researcher who applies for them. So I would encourage people who are seeing families with autism that these studies are available, as well as Ian, to make sure they know about these resources to help recruit families. I think the other important thing to think about, which all of us are involved in, are the clinical areas. So we found just through our cytogenetics laboratory that we're identifying a lot of new causes or new imbalances in children with autism, which provide great opportunities for identifying new regions that can be implicated in this syndrome. And what we've tried to do is build a cytogenetic consortium of clinical laboratories. It wasn't started for research purposes or to deal with our interest in autism, but it was mainly started because of all of the clinical laboratories starting to offer array analysis and and the very differing types of arrays that they were offering. We thought there needed to be some type of standardization so that clinicians, genetic counselors, and families weren't being confused about what test was being run in a genetic laboratory. So really the main goal of the consortium was to standardize the design of a cytogenetic array and also standardize data interpretation so that labs weren't calling things of different clinical significance. But a great side effect is that we do have a large number of samples that are being analyzed every week. So now there's more than 70 labs that have joined this consortium, which is called the International Standard Cytogenomic Array. It was a play on ISCN, which is cytogenetic nomenclature. But we estimate that in these laboratories that at least 25,000 clinical cases have been analyzed in the 2 years since we've been established, and current caseload is about 500 cases per week. So it's a great resource for looking across cases between different laboratories and seeing, are we finding any new recurrent rearrangements that could key us into looking for new causes of autism. This is just a picture of the type of array that we use that has whole genome coverage, and then we have targeted coverage across regions of the genome that we already know are associated with certain phenotypes. And the resolution really differs on the array that's being used, and there are various formats ranging from 44,000 oligos to 180,000 oligos, and some of the other platforms like Affy and Illumina have up to 1 million oligos, but still looking for copy number variation across the genome. And as was mentioned, one of the newer regions that's been linked to autism is 16p11.2, and in particular, deletions of this region have been linked to autism. I think with duplications, the jury's still out a little bit. The duplications have been observed in normal populations, but they seem to be enriched in families with autism. So I think more studies need to be done in larger numbers for the duplication, but clearly the deletion now, I think, has enough data to support a link to autism and a syndromic phenotype. And this first study was actually done by using agree samples as well as other autism cohorts, and one of which was the data from the Boston Clinical Lab. This just shows that they got good press with being in the New York Times and saying, we found a new cause of autism in a 16p11.2 deletion. These are two unrelated children who you can see have similar facial features. This just shows an example of a microarray result. So here's all the normal oligonucleotides, but here you can see the shift to the left, indicative of a deletion of this region. And the deletion is about 600 kb in size and has about 29 genes in the region. And when we looked at our laboratory data, we found that just within Emory that we had about 13 cases that had deletions, including this region. And the gene content is shown down here. When we polled the other laboratories, and this was actually just a small portion of the other laboratories that we knew had larger case numbers, we actually found that there were 22 cases of the same region of deletions from other labs. So from the total number we looked at, there were about 35 out of 10,500 cases, or about 1 out of every 300 cases analyzed in a clinical lab has a deletion of this region, which corresponds to about 1 to 2 cases being identified in just a small subset of our labs. Most of them have been de novo. There's been a few inherited, but I think it's important to note that most of them that are inherited have been the parent. When you go back and look, they say there are some mild cognitive issues. So not totally normal psychological phenotype or cognitive phenotype. And the Boston group has done a lot to start to do early phenotypic assessments of this. And most of the children have fall somewhere on the autism spectrum disorder trajectory. They have developmental delay, some have MR and delayed speech. Most have hypotonia early on. They have the mild dysmorphic features. A lot of them have problems with weight, and as far as behavior, they seem to be very anxious and sometimes aggressive. So they do have behavioral problems that need special attention. And actually the Simons Foundation has become very interested recently in trying to take individuals with known genetic causes and follow them up to try and define more what the consistent clinical features are, and are there certain treatments, even behavioral treatments, that might be more successful in children with, say, 16p11.2 deletions versus Fragile X. So is ABA better suited for people with 16p11 than Fragile X? Are there speech therapy that maybe these individuals need that they don't? So really trying to take what we know about genetics and tailor treatments to these individuals. These just show our overall case numbers in looking at cytogenetic abnormalities, and we've done more than 3,000 array cases in our lab to date, and that's everything, including developmental delay, MR, and ASDs. And we have about a 20% yield for pathogenic imbalances. We looked at how many we knew had a true known diagnosis of autism, so they had ADI or ADOS or something. So, and of those, there were about 370. And when we look at those 370, we see that 32 of them, or about 9%, have pathogenic abnormalities. So still, around 10%, which is a good yield for finding a known cause of a disorder. What we're starting to do now is take these positive pathogenics and do data curation. So look to see, do we have overlapping cases? Are there single cases that maybe just involve very interesting candidate genes? And so I'm going to show you in the next few minutes a few examples of those. So one of the regions that we found to be recurrent in our autism population are deletions of 17q12. And this shows the array result with a deletion. Here you can see all the oligos shifted. It's about a 1.5 megabase deletion. It includes 15 genes. And it's not a new region. So this region had actually been previously reported as a recurrent microdeletion, so mediated by segmental duplications, just like DeGeorge syndrome, by Medford et al. From Evan Eichler's group. But they reported it in association with renal disease or diabetes. And there is one gene in this region. There's one gene in this region, TCF2, which has been associated with maturity onset diabetes of the young. And what they had done was screen patients with MODY5 to look for these deletions and found them. They also found these deletions in a population of stillbirths that had kidney abnormalities. But no report of neurodevelopmental issues was noted in these, and they actually made a point to say that there weren't neurodevelopmental abnormalities. So in our cohort from the clinical laboratory, we actually identified 5 males and 1 female with deletions of this region through microarray testing. And so when we look at these, the males, 4 out of 5 males were referred for autism. 1 male was referred for developmental delay, and his autism assessment is currently pending. Of those, 4 out of 5 were de novo. We had 1 that was inherited from a mother, but the mother did not have a normal cognitive phenotype. So she has bipolar disorder and was noted to have mild cognitive delays through her lifespan. The other female that we found was not referred with a diagnosis of autism. She was referred for mild cognitive delay and speech delay. This was a de novo rearrangement, and in speaking to her physician, also does not feel that she fits on the autism spectrum yet. When we went back and actually got consent to study these patients as part of a research project, we obtained pictures and started to try and look for common features. We do feel like there are some common dysmorphic features, which I'll show you in a second, but most have, or all have macrocephaly. They all have some mild to moderate level of developmental disabilities. They have nail abnormalities, and different degrees of kidney anomalies were noted, some of which were noted on ultrasound and others which weren't noted till later in life. None of these individuals have signs or symptoms of diabetes, but most are too young to be assessed, or that you would expect to see that. And these are just pictures of the cases that we've recruited, or the cases that I have permission to show so far. So these are 6 cases. These are our, this is case 3 is actually the mother of case 5. So this is our female and 4 of our males. And we actually were pretty surprised. I don't think this is a recognizable phenotype that when you walk into the clinic you would pick up, but looking at these pictures all beside each other, I think there are some features that sort of stand out. So when they're younger, they sort of have more of a bulbous nose, they have a little bit arched eyebrows, epicanthal folds. But we also notice when we have pictures that go across different ages, we sort of notice a transition in their faces. And so this is the same individual, and this is the same individual, and this is the same individual. In that, I mean, their forehead is very prominent. You can see that in some of the side views. But their nose also goes from sort of being short and bulbous to, by teenage years, getting longer and extended. And the one picture that we can't show actually is a 17-year-old who they really just look strikingly similar. So it's not something I would say when they walk into clinic, you should say this person has a 17q12 deletion, but you might think of it with kidney abnormalities. So it might be something, autism and kidney abnormalities, to think of this as a differential. Another interesting aspect of this, and what one of the regions Brad pointed out in his talk, was that there have been linkage and association studies that have focused on 17q. And one of these actually showed that there was evidence for specific risk alleles in autism spectrum disorder. And that region overlaps with this 17q and 2q deletion, so it's very close with that male only. And so we think that suggests there could be a specific risk allele for autism in this region. And interestingly, as I pointed out, all of our male patients had autism or autistic features, and thus far the females we have do not. So strongly suggesting that the 17q12 microdeletion could be associated with an increased risk of autism, especially in males who have this deletion. And this just is a summary of some of the established and new recurrent microdeletion duplications and their relative frequency, which I think is interesting. We call these rare disorders, but collectively they're actually quite common. And this data was actually compiled by John Barber, who's one of the members of the ISCA consortium from Salisbury in the UK. He took our data on about 2,400 samples and looked at the frequency of abnormalities we found in these recurrent microdeletion syndromes, and then estimated what the population frequency would be. And you can see in the two I mentioned, 16p11.2, it's about 1 in 173 in our cases, or 1 in 4,500 in the population. 17q12 is about 1 in 350 in our cases, or 1 in 9,000. But when you look at all of these collectively, you can see that these recurrent microdeletions, so not even counting the other deletions and duplications that occur randomly throughout the genome, occur in about 1 in 26 cases that we test in the clinical lab, which translates to about 1 in 681 in an estimate of population frequency. And if you compare that again, like something like cystic fibrosis, you can see these actually are common as a collective group. And then the last example I'll show you is one interesting area that we've been trying to focus on, and that's imbalances that we find that only involve 1 or 2 genes. So de novo imbalances in affected proband, where we think, if you have limited that deleted region to 1 or 2 genes, that gives you a really strong candidate for trying to look for do mutation analysis in other individuals who have idiopathic autism. This is one example that is a deletion of the short arm of chromosome 3. This was done in collaboration with the University of Mississippi, where there's a deletion that's about 1 megabase in size. And here, this shows the genome browser view. This red bar is our deletion, and these are the known genes. A lot of these lighter genes are hypothetical proteins, but this is a known gene, FOXP1. And so in this patient with a de novo deletion, that was 1 gene, there's only 1 gene deleted. And this is interesting to us because FOXP2 deletions have already been shown to be reported in individuals with speech delay. And this particular patient presented to the genetics clinic with speech delay, mild motor delays, and KRE1 malformation. There's been another report in the literature recently of a deletion of 4 genes of 3P that included FOXP1. And this individual also showed speech delay along with other congenital malformations that were much more severe than our case. But we think that this is building evidence that FOXP1 deletions are linked to speech and language delay. And when you look at a pathway analysis to start to figure out how does FOXP1 and FOXP2 relate, they're actually quite interrelated. So that FOXP2 is here, and you can show a direct interaction with FOXP1. But looking at this type of analysis also allows you to see what other genes you might want to look at as candidates for speech and language development. So here we see that another member of the FOXP family, FOXP4, interacts closely with these. And then interestingly, FOXP2, you can see interacts with this contactin-associated protein 2, which has already been associated with autism, and particularly with some of the language developments you see. So by using some of these single cases, you can start to tease out good candidates for other research protocols to look at some of these specific endophenotypes. So in conclusion, we're showing that whole genome cytogenetic array analysis is enhancing the diagnosis of ASDs, and we actually use this as a first-line test now. So we recommend array before chromosome analysis. The examples shown demonstrate the utility of having a high number of cases available to you to look for common abnormalities that occur. It also, I think, gives us an estimate of what the frequency is in these populations. And then even looking at rare cytogenetic abnormalities can help us to discover new genes linked to these disorders. I'd just like to finish by thanking my collaborators. So at Emory, David Ledbetter, who is a research collaborator of mine and our division director. Erin Baldwin-Kaminsky is the research manager, laboratory manager for my research laboratory and does a lot of our bioinformatics work. Katie Rudd and Mike Rossi are two other co-laboratory directors who sift through all the array analysis for the clinical testing with me. And then Daniel Moreno-DeLuco is the postdoc in my laboratory who's done all the work on the 17q12 phenotype and is writing that up. I'd also like to acknowledge our collaborators from the University of Mississippi, Omar Abdul-Rahman and Christopher Carr and Holly Zimmerman, the genetic counselor, who were instrumental in getting the FOXP1 paper together and submitted. And then just all the, of course, our grant support, and then all the families and physicians and genetic counselors who always listen to our phone calls and try to get us samples for our research studies. So thank you very much.
Pam Martin, Jacksonville, Florida. I wondered if you could comment on, I think, an off-the-cuff comment you just made about the utility of chromosomes versus array.
You caught that me saying that?
Yeah, you did. And that's something we've been talking about. I mean, should we just be going to starting with array CGH? And is there, what do you think about it? Because you're going to miss the balanced translocations, but you're looking for abnormalities, not.
Right. Yeah, no, and we, for a long time, said you could order both, and what really made us make the decision is some of the insurance companies are saying you can have one but not both, so which one would you choose? And we picked array. Because I think even the balanced rearrangements, most of the studies that are looking at individuals are apparently balanced rearrangements. Most of the studies that take individuals with abnormal phenotypes and look at those, there's actually some deletion or duplication of the breakpoint or something like that. So we feel like you're getting a twofold yield at least out of doing array first. And if you looked at doing karyotype and how much you miss, it's actually a disservice to your patients to make them wait for that extra test, go through a normal test and then have to go through array. So we actually recommend that now. Brad?
Yeah, so the numbers you showed with those 3,000 patients, would you guys publish that, please?
Yeah, well, it's published, it just isn't published in that manner. So we published a paper in Human Molecular Genetics on, we took our data and just looked at how many of our cases were mediated by segmental duplications, how many have been reported, and how many new ones did we find. And John Barber took that data and put it into that nice frequency table, which we were very impressed with. But yeah, we could do that just as a follow-up to that paper.
Yeah, because it's just, I mean, it's nitpicking, but it's not. Again, this whole idea of evidence-based medicine,
Yeah.
that's not in the literature. Most of us have come to the same conclusion you have, which is that in reality, microarray should be a first-year test.
Right.
But there's not the literature published to
Right.
support that yet. So many of us are making that practical decision.
Right.
But believe it or not, a lot of the medical directors of insurance companies read the literature occasionally. And when that's listed not that way, then oftentimes that I would much prefer to do the microarray first, but you actually have the literature not supporting it.
Right.
So it'd be nice, because our little 90 patients isn't anywhere close. It'd be nice to have that kind of thing in the literature.
Right, and we actually, as part of the consortium, that was one of our first goals because we thought we needed to get the literature out there. So David Miller from Boston Children's has it's pretty close finished draft that's pretty much the recommendation, and we've talked to other groups about doing the same, but it's a good idea to publish that as a frequency table.
I would, yeah.
Because we didn't take it and change it like that in our paper, and so it's nice that it's there now.
Well, along those lines, maybe it's time now to bring this to the committees, the cytogenetics committee at the College of Medical Genetics with Society, and have them pass off on some clear guidelines.
Right.
I don't know if you're associated with them, but that
Yeah,
would be
there's a microarray committee that we did mainly for the testing side of it, and a little bit of interpretation and how to do proficiency and how to validate. But yeah, I don't know that there's yet from the clinical side saying do array first.
But I think we were all seeing enough data now with the common use of microarray that the topic's mature enough to at least discuss in that forum.
Yeah, I think that's a good suggestion.
our speakers one more time. We are ready to adjourn unless there are any more.
I'm going to say that.
We're ready to adjourn unless there's any more announcements, and we will see you in Jacksonville next summer. Have a safe trip home.